Okay, good afternoon for most of you. Welcome back. This is our test to see what we got out of yesterday's test prep. For anyone that's here for the first time, this is content that we covered yesterday in our webinar that we're now just assessing and making sure we got it. But feel free to participate nonetheless. We will have about 40 seconds per question. I encourage you to put your answers in the chat box. If you are coming at me from Zoom, your answers are confidential. Only I see them. If you are out on our other social media platforms, people will see your answers, but I still encourage you to take a chance and get that answer out there. That active participation certainly helps our brain stay engaged. It will be very fast paced. I believe that I have about 19 questions planned, and our time really is about an hour and a half. So we're going to try to get through them as quickly as possible while still covering what you need to know. We will have the question in front of us, about 40 seconds to answer, and then our next slide will reveal the answer, the rationale as to why it is the answer, and then the rationale as to why the others are incorrect. And so that will be our format for today. But before we get started, I do like to always remind you of all the wonderful things that we have here at TEAS to support, at TEAS, at Archer Review to support your TEAS studying. Let's get that straight. So as you know, all of our webinars are free, and that is that top QR code. You can rewatch old webinars. That's our second QR code. So if you can't always attend at the times that we have them, that's a great way to participate. We also have our question bank, which has a free trial, and then it does cost depending on how much time you would like to have access to it. And our content here says it's over 1,500 questions, but we've been working diligently on that. And the last I checked, it was actually over 1,700 questions with rationales. So it's a fantastic opportunity, and you can go in and you can customize it for areas that you need to work on. So let's say for like today, for example, let's say that you get all the macromolecule questions wrong. You would know that those were the ones you wanted to focus on in our question bank, and you could customize it for that purpose. So it's a really cool option. We also offer on-demand videos. So as you know, if you've been to any of our other webinars, the webinars give you a taste, give you an overview, but they don't get into the nitty-gritty. The nitty-gritty is in our on-demand videos. That really gets into the deep content. We also have tutoring available. Myself and two other tutors are available to cover all of your TEAS needs. And then we have two packages. We have the SurePass package and the SurePass Pro package. Differences really between these two is that the SurePass gives you pretty much everything you need except for private tutoring, and the SurePass Pro gives you an hour of private tutoring. So check those out as well. And then, as always, you can reach us on all the socials, Facebook, Insta, TikTok, and even emailing us at prenursing@archerreview.com is an option. As I said, you have 3 tutors to satisfy all of your preparatory needs for the TEAS. My colleague and friend, Tammy, she does English and reading. You can scan that QR code there. I am Shellie Harshberger. I do the science. You can scan that QR code if you'd like to have me as your science tutor. And then you've got Brandon. He is our math expert. So feel free to scan those QR codes or access our website and request a tutoring session. Okay, so we are going to kick off our first group of questions focused on macromolecules. So let's get started. Which of the following correctly matches the carbohydrate structure, type, and function? A, monosaccharides, sucrose serve as an immediate energy source. B, disaccharides, glycogen is stored in the liver and muscles for energy. C, polysaccharides, glycogen is stored in the liver and muscles until needed for energy, or D, monosaccharides. Glycogen is the simplest form of carbohydrate used for energy. All right, you got about 40 seconds. Get those responses in there. Okay, let's take a look at our answer. So our correct answer was C. Glycogen is a polysaccharide stored in the liver and muscles until the body needs energy. When required, glycogen is broken down into glucose, and that's what helps maintain our sugar levels and our energy levels. The reason answer A is incorrect is because sucrose is a disaccharide, not a monosaccharide. The reason answer B is incorrect is because glycogen is a polysaccharide. Oops, didn't mean to start that over again. Glycogen is a polysaccharide, not a disaccharide. And then answer D was incorrect because glycogen is a polysaccharide, not a monosaccharide. So we want to just make sure that we're paying close attention to what the category is, what the specific type was, and then its function. So C, polysaccharide, glycogen, those line up, and then its function is that it's stored in the liver until it's needed for energy. All right, our next one. Which of the following are characteristics of the plasma membrane? Now, this is a select all that apply, so make sure you are attentive to that. A, it is composed of a phospholipid bilayer with hydrophilic heads and hydrophobic tails. B, the hydrophobic tails face outward while the hydrophilic heads face inward. C, it functions as a selectively permeable barrier, regulating the movement of substances in and out of the cell. D, the phospholipid bilayer allows both water-soluble and fat-soluble molecules to pass through freely. And E, the arrangement of phospholipids helps support the plasma membrane's function. You got about 40 seconds. I'm seeing some good answers out there. Read thoroughly. There are several correct answers here. Okay, let's take a look at our answers. So the correct answers were A, C, and E. So let's take a look at those before we look at our rationale. So A, the phospholipid bilayer and hydrophilic heads. C, selective permeable barrier. And E, they help support the function. So let's take a look at the details. So A is correct because the membrane is composed of that bilayer that we talked about yesterday. And we've got those heads that face outward that are fond of water. They like water. And we've got the tails that face inward because they are hydrophobic. They are not a fan of water. C is correct because the membrane has the ability to determine what can enter and exit the cell. And then the structure of the phospholipid bilayer is what helps provide support and cell protection and regulates what enters and exits the cell. So that's why E is correct as well. Now let's take a look at why B and D are incorrect. So B is incorrect because the hydrophilic heads face outward towards that watery environment and the hydrophobic tails face inward, not the other way around. So just remember that those ball looking things, the heads, those are on the outside and the tails align on the inside. And then answer D is incorrect because the phospholipid layer allows small non-polar molecules in and out of the cell, but it restricts molecules that are water soluble or unless they're transported through membrane proteins. Okay, this is another select all that apply. Which of the following statements about enzymes are true? A, enzymes lower the activation energy required for reactions to occur. B, enzymes are consumed during chemical reactions and must be replaced. C, enzymes bind to specific substrates in a lock and key fashion. D, enzyme activity can be affected by temperature and pH. Or E, enzymes speed up chemical reactions without being used up. Get those answers out there. Okay, let's reveal our correct answer. So our correct answers, they were all except for B correct. So let's look back again real quick. So they lower activation energy, they bind in a lock and key fashion, they're affected by temperature and pH, and they speed up chemical reactions. So let's look at our rationale. So A, they're lowering that activation energy so it makes chemical reactions occur easier and it doesn't take as much energy, which is more energy efficient for our bodies. Yesterday, we talked about how they fit in this lock and key fashion. So they attach to what's known as their substrate very specifically. D, we talked about yesterday as well, that enzymes, the one con to an enzyme is that they are easily impacted by temperature and pH. And if they are exposed to a temperature outside of their range, or if they are exposed to a pH outside of their range, they do something known as denature, which just means that they change shape, they kind of break down. And at that point, then they're not functional and they're typically sent to the lysosome to be broken down and expelled from the cell. As far as E being correct, they act as catalysts. So catalysts and enzymes are almost used synonymously. That means that they are going to speed up reactions, but they don't get used up. And that is a big benefit to an enzyme because they can just go on and catalyze reaction after reaction after reaction. So that's what makes B incorrect, is that they are not consumed in their reactions. They can go on and help assist other chemical reactions. All right, our next one. Which of the following correctly describes the structure of DNA? A, DNA is a single-stranded molecule composed of amino acids. B, the rungs of the DNA ladder consist of sugar and phosphate molecules. C, DNA has a double helix structure with nitrogenous base pairs forming the rungs. Or D, in DNA, adenine pairs with cytosine and guanine pairs with thymine. Get those answers out there. Okay, let's see what our correct answer is. Our correct answer is C. So let's take a look. DNA has a double helix structure with nitrogenous base pairs forming the rungs. So if we remember our picture from yesterday, I pulled an excerpt here for us. We have our double helix structure. That's what's illustrated on the right. And then when we look, remember that our sides of the letter were our sugar and phosphate. And then the steps of the letter are base pairs. And then remember, it's hydrogen bonds that bond them together. Okay, so let's take a look at why the other answers were incorrect. So answer A was incorrect because DNA is double-stranded and it's not single-stranded, which is what A stated. And it's composed of nucleotides, not amino acids. Remember, amino acids are proteins building blocks, not DNAs. And then the rungs of the ladder are composed of the nitrogen bases, while the sides are the sugar phosphate backbone. And then the pairing for D was incorrect. A pairs with T and C pairs with G. Now the pairing I use kind of the structure of the letter to help remember which is correct. A is a very pointy, got points to it, sharp kind of letter. T has points to it, where C and G are very curvy. And so I kind of line them up by how the letters are structured to remember that G pairs with C and A pairs with T. I did have a question that asked me, someone said in the chat if they got some of the correct answers on the TEAS, how would that be scored? And I do believe that there is no partial credit given. I believe that it's either an all or nothing kind of thing on the TEAS. So that is my best knowledge. Obviously, I am human. I could be wrong, but I believe from the last I understood it, you have to get all of the correct answers. Another person just asked what rung means. It's another word for steps on a ladder. So like T plus A down here on the bottom, that would be the first step or the first rung of the ladder. I'm not sure why we refer to the steps on a ladder as a rung, but it's kind of a common term when thinking of the steps on a ladder. So great questions, guys. Thanks for putting those out there. Okay, we are now moving into our next category, which is cell structure and function. What is the primary difference between vacuoles and lysosomes in eukaryotic cells? A, vacuoles store materials while lysosomes break down waste and cellular debris. B, lysosomes store nutrients while vacuoles are responsible for digestion. C, vacuoles contain digestive enzymes while lysosomes store water and nutrients. Or D, lysosomes are only found in plants while vacuoles are only found in animal cells. Get those answers out there. Hey, our correct answer was A. So let's take a look at that. Vacuoles store materials while lysosomes break down waste and cellular debris. So we talked about this a little bit yesterday. Our vacuoles are primarily used for like water storage, things like that, nutrients and waste, where lysosomes actually have enzymes in there that help break down. Like I talked about earlier, if an enzyme is denatured, it gets sent to the lysosome to be broken down so that it can be exposed or expelled from the cell. I referred to them yesterday. I struggled with coming up with the term, but I referred to them as kind of the garbage disposal of the cell because they can use the enzymes to break down old organelles, cellular debris, denatured proteins, and then those substances can be expelled from the cell. To look at our other rationale, lysosomes do not store nutrients because they're the garbage disposal. Vacuoles will be storing nutrients, so that would be a great one to help compare and contrast those. On the flip side then for answer C, vacuoles are not digesting anything. They're not the garbage disposal. They're more of a storage location. You could think of the vacuole almost as like your pantry. And then D, plants do have a large central vacuole. That's where their water goes when we water them, and that's where they access the water between waterings. where we have lots of smaller vacuoles. And then we have lysosomes in our cells, but plants don't typically have lysosomes. All right, our next one. What is the primary difference between the nucleus and the nucleolus in a eukaryotic cell? A, the nucleus produces ribosomes, while the nucleolus stores genetic information. B, the nucleolus is responsible for storing DNA, while the nucleus assembles ribosomes. C, the nucleus controls cellular activities and contains DNA, while the nucleolus produces ribosomes. Or D, the nucleolus regulates gene expression, while the nucleus synthesizes proteins. Let's get some answers out there. I'm seeing some good ones. All right, let's take a look. Correct answer is C, and I think almost every single person got that correct, so well done. So what was our correct answer? That the nucleus is in charge of cellular activities. It's where our DNA is located, while the nucleolus is producing ribosomes, okay? So keep that in mind. And remember, I talked about yesterday how be attentive to the word that has Leo in the center of it is the structure that's in the center of the nucleus. So be attentive to that. As far as our others, why they're incorrect, because the nucleus for A is where our genetic information is stored. The nucleolus's job is building the ribosomes, and it had it backwards. For B, there is not DNA stored in the nucleolus. The DNA is in the nucleus. So that made that incorrect. And then for D, the nucleus regulates gene expression, but proteins are made on the ribosomes, not the nucleus itself. So that's what made that incorrect. All right, our next category is microorganisms. Which of the following correctly differentiates between microscopic animals, protozoans, and fungi? Select all that apply. A. Protozoans are single-celled eukaryotic organisms, while microscopic animals are multicellular. B. Fungi and protozoans are both eukaryotic, but fungi act as decomposers or parasites, whereas protozoans are primarily free-living or parasitic. C. Helminths, such as tapeworms and roundworms, are protozoans that cause intestinal infections. D. Malaria is caused by a protozoan from the Plasmodium species, which infects the liver and red blood cells. E. Fungi are prokaryotic microorganisms responsible for infections such as athlete's foot and ringworm. Get some answers out there. Okay, before we go through the answers, I did have a couple questions I wanted to address. Someone asked what synthesis means or synthesize means, and I'm so glad you asked that. In science, when you hear the word synthesize, all it really means is to make something. But for whatever reason, science likes to use that word synthesize. So anytime you hear that word synthesize, it means to make. For example, one of the categories on the TEAS is protein synthesis. What does that really mean? It just means this is how the body makes proteins. All right, so getting back to our microorganisms here, let's take a look at what all the correct answers were. A, B, and D were our correct answers. So protozoans are single-celled eukaryotic organisms, while microscopic animals are multicellular. That is correct. Okay, that was our first one. And it's telling us protozoans are single-celled eukaryotic organisms, while microscopic animals, those helminths, are multicellular and they can live in our intestines. And remember, I talked yesterday about how if they live in our intestines, they take away our nutrition and they can get stuck. And if they get stuck, then we have the inability for waste to move through our body, through nutrients to move through our body. It's a real problem. Let's take a look at B. So B is also correct. Fungi and protozoans are both eukaryotic. Remember, those are complex cells. But fungi can be decomposers like we see in nature, or parasites like ringworm, where protozoans are either free-living or parasitic. Let's see what else it has to tell us about it. So yes, both are eukaryotic. Fungi are going to be decomposers or parasites, but then protozoans will either be free-living or live as a parasite in something, but they are single-celled. Remember, we talked about how they are one of the unique organism categories that actually are made of one eukaryotic cells, or one eukaryotic cell, which is unique because most eukaryotics are multicellular. Our next one then, D, malaria is caused by a protozoan from the Plasmodium species, which infects the liver and red blood cells. So yes, if you remember, we talked about yesterday, I even drew an X over a picture of the mosquito because I wanted you to not think that a mosquito was a protozoan, because it's not, but they carry the protozoan in their blood and in their body. And what they actually do is when a mosquito stings you or bites you, whatever you want to call it, they inject a little bit of a numbing agent so that you don't feel them pulling the blood from your body. And within that numbing agent is where this protozoan lives. And so that's how you can get malaria. And that is something that's going to then invade your red blood cells. It infects your liver and is, life-threatening. Now, the other ones that were incorrect, why they were incorrect. Helminths, both tapeworms and roundworms, yes, they're microscopic animals, but they're not protozoans. That was a whole different category. They are multicellular parasitic worms. For E being incorrect, it's because fungi are eukaryotic. They are not prokaryotic. They have complex cells like us. Okay. So let's see, someone's asking me, protozoans are microscopic animal or not? So protozoans are microscopic single-celled eukaryotic organisms, okay? But they're not a microscopic animal. That would be our helminths, our tapeworms, things like that. But they are single-celled eukaryotic organisms. Okay, moving on. Which of the following correctly describes the differences between gram-positive and gram-negative bacteria? Select all that apply. A, gram-positive bacteria have a thick peptidoglycan cell wall, while gram-negative bacteria have a thin one. B, gram-negative bacteria stain pink because their thin peptidoglycan layer does not retain the crystal violet dye. C, gram-positive bacteria stain pink due to their thick peptidoglycan wall. D, antibiotics are often designed to target structural differences between gram-positive and gram-negative bacteria. Or E, gram-positive bacteria stain purple because their thick peptidoglycan layer retains crystal violet dye. Take a look at those. I'll get a timer going. All right, let's take a look at our correct answers. So 4 of these were correct, A, B, D, and E. So let's take a look at A first. So A was gram-positive bacteria have a thick peptidoglycan cell wall, while gram-negative bacteria have a thin one. So this tells us that yes, that's what we're looking at. Gram-positive has that thicker wall, whereas gram-negative has the thinner wall. Then B, gram-negative stain pink because of that peptidoglycan layer and does not retain the crystal violet dye. And that is correct. Gram-negative bacteria stain pink because that thin peptidoglycan layer doesn't hold on to the violet dye. It actually instead holds on to the counter stain, which is the, I don't know how to say that, safranin. There we go, safranin. I haven't had to say that in a long time, been a long time since I've stained bacteria. All right, our next one that was correct was answer D. Antibiotics often target these structural differences, and that is very true. And that is why, as a nurse, you will need to understand the difference between gram-positive and gram-negative bacteria, because the antibiotics that will be prescribed often target those structural differences. So as this tells us, for some example, antibiotics target the synthesis, remember that means to make, so they target the making of the peptidoglycan. And so that actually affects gram-positive bacteria then more than gram-negative. And so that's something that's important as a nurse to understand, as those bacteria or those antibiotics will be tackling that cell wall. And then finally, answer E was correct. And answer E was saying that the gram-positive stain purple because of that thick peptidoglycan layer, and they hold on to that violet dye. And that is the accurate response. Because of the peptidoglycan layer, it's really attracted to that violet dye. And so then answer C was incorrect because gram-positive bacteria is going to stain purple, not pink. It's gram-negative that stains pink. Okay. And again, that has to do with that peptidoglycan layer. Okay. All right. Moving right along here. Why are viruses classified as non-cellular entities? A, they have a nucleus but lack organelles. B, they cannot reproduce or carry out metabolism without a host cell. C, they contain both DNA and RNA within their capsid. Or D, they have a plasma membrane but lack a cytoplasm. Get those answers out there. So before we go over the answer, I had someone ask, how can we tell this difference between the gram-positive and the gram-negative bacteria and staining purple or staining pink? And that's a great question. And what I try to remember is that gram-positive means it has more. It has more of that peptidoglycan. And when you'll be in the lab at some point in school staining bacteria. I think it's kind of a natural rite of passage for nurses. When you get the stain to stain the bacteria, it is purple. It is a really dark violet purple. It almost looks like you're getting a vial of ink. And the purple is attracted to that peptidoglycan layer. Okay, so the more peptidoglycan, the more purple. So think positive equals more equals we're going to see purple. It's going to be just totally attracted to it. Where if there's not a bunch of peptidoglycan, the stain's just not going to be as attracted. And actually, a counter stain is what ends up being attracted to it. But if you just really focus on the purple, then you know that the other is going to be the negative, or the one that doesn't have as much peptidoglycan in its wall. So I hope that helps. That's kind of how I stick it together in my brain. All right, so back to our viruses. Let's take a look at our correct answer. Our correct answer was B. So B says that viruses cannot reproduce or carry out metabolism without a host cell. Okay. So that's why they're classified as non-cellular, and they even don't say that they're truly a microbe. They call them entities because they cannot function without hijacking our cells. I used to teach high school too, as well as college level. And I used to tell my high school students that if we could really see viruses, we'd be terrified of them because it's as if they just are like the brain sucker. They attach to our cells and they completely like zombify our cells. They have the ability to completely take over our cells. And whatever that cell was doing before the virus attaches to it, it's done. Now its entire job is to make new viral entities. So they're actually a really creepy little entity. Looking at our other ones, they have a nucleus but lack organelles. No, viruses don't have nucleus, they don't have organelles. Instead, they just have genetic material, either DNA or RNA. And then they're typically wrapped in what they refer to as a capsid, which is a protein coat. I'm looking at C. They contain both DNA and RNA within their capsid. The reason that's incorrect is because they'll contain one or the other, but not both. So they will have either DNA wrapped with that protein coat or RNA, but they won't have both. Then for answer D, they, excuse me, they don't have a plasma membrane. They don't have cytoplasm. Again, they just have this protein coat. Every once in a while, a virus will pick up a kind of a lipid envelope that they got from a host, but that isn't even something that was theirs. They kind of picked it up from the host. Okay, we are moving on to our next category, which is called cellular processes. Which of the following statements about DNA replication are true? Select all that apply. A, DNA helicase unwinds the double helix and breaks the hydrogen bonds between bases. B, the entire DNA molecule is copied into RNA during replication. C, each original DNA strand serves as a template for a new complementary strand. D, DNA replication produces two identical DNA molecules, each with one old and one new strand. And E, the newly formed DNA molecules contain two entirely brand new strands. Get those answers out there. Hey, let's see what answers we got. So our correct answers are A, C, and D. So let's look at A. So A, DNA helicase, remember that's an enzyme, unwinds the double helix and breaks the hydrogen bonds between the bases, okay? So it unwinds them, it's breaking those hydrogen bonds, and it allows the process to start. Answer C, each original strand serves as a template for the new complementary strand. We see that here. I brought this picture over from last night's practice session or our test prep. So we see that our original DNA here, we've got a green strand of that original DNA, in the newly formed strand. We've got our blue strand here with the new strand forming on it. So each strand serves as a template for the new one. So we have one old and one new strand. All right, looking at answer D. Answer D, DNA replication produces two identical DNA molecules, each with one old and one new strand. And that's what we were just talking about there. Okay, so now let's take a look at B. What was wrong with B? The entire DNA molecule is copied to RNA during replication. The answer is incorrect because DNA replication copies DNA into DNA, not RNA. That was the issue there. And then E, the newly formed DNA molecule contains two entirely brand new strands. So the reason that's incorrect is what we were just talking about here, how we have one old strand and the one new strand forming. Okay. All right, I've got a couple questions here. A few times I've seen the word abide, and I'm not sure what's being asked there. I'm so sorry. Please elaborate on what you mean by that. And then someone else asked if gram-negative is pink or red. No, it actually stains pink. It is not a red. It has to do with the counter stain to that violet. Okay. All right, moving on. Which of the following statements correctly describe the processes of transcription and translation? Select all that apply. A, transcription is a process where RNA polymerase produces mRNA from a DNA template. B, during transcription, adenine in DNA pairs with thymine T in mRNA. C, translation occurs in the nucleus, where mRNA codons are read to assemble proteins. D, transfer RNA carries amino acids to the ribosomes, matching codons with anticodons. Or E, codons in mRNA are a group of 3 nucleotides that correspond to specific amino acids. Get those answers out there. Okay, let's take a look at our responses. So our correct answers are A, D, and E. So A, transcription is a process where RNA polymerase produces mRNA from a DNA template. Okay, so that's reminding us that transcription is taking place in the nucleus where that mRNA strand is being made. And if you recall, I said yesterday that essentially I like to think of it as the recipe book for those proteins is locked in the nucleus. It cannot leave. And what I mean by a recipe book is DNA. And so the only way we're going to get the instructions to make that protein is to make a copy of it. And that copy is essentially mRNA. All right, D was correct. So D, transfer RNA carries amino acids to the ribosome, matching codons with anticodons. So yes, tRNA is our transfer RNA. It's carrying over amino acids. Those are our building blocks of the proteins. It's carrying them over the ribosome. And how it knows it has the right amino acid is that it has 3 bases on the bottom of it that it lines up with mRNA. So A pairing with U, G pairing with C, and so on. And so if its bases line up, then it knows, yep, I've brought the right amino acids, and it drops it off. I often, again, like I said, I taught high school and would teach this, and I would tell my high school students that it's kind of like FedEx driver. He has to make sure he's at the right address. And in this case, it's the anticodon lining up with the codon that tells it it's the right address, so to speak. All right, E. Codons in mRNA are groups of 3 nucleotides that correspond to specific amino acids. So yes, so codons are a set of 3. That's that address that I was kind of using as an example. And that set of 3 correspond to amino acids. And that's what's telling tRNA, yes, you've got the right amino acid. You can drop it off so that we can continue building this polypeptide chain that's going to form our protein. Okay. All right, now why some of the others were incorrect? B, during transcription, adenine in DNA pairs with thymine T and mRNA. Remember, no, RNA has no thymine. None of the RNA has thymine. It has uracil instead. So that's what it's telling us here, that adenine's going to pair with uracil, not thymine, because thymine's only found in DNA. And then translation occurs in the nucleus where mRNA codons are read to assemble proteins. No, translation occurs at the ribosome. So everything else is pretty much correct here, but the location was wrong, okay? Translation is occurring in the cytoplasm on the ribosome, okay? All right, moving on to our next one. Which of the following correctly contrast mitosis and meiosis? Select all that apply. A, mitosis produces 2 genetically identical cells, while meiosis produces 4 genetically unique cells. B, mitosis occurs in germ cells, while meiosis occurs in somatic cells. C, meiosis involves 2 rounds of cell division, while mitosis involves 1. D, the daughter cells in mitosis have the same number of chromosomes as the parent cell, while meiosis halves the chromosome number. Or E, both mitosis and meiosis produce gametes for sexual reproduction. Let's get those answers out there. All right. Before I expose the answers, I had out on one of our socials someone share something really cool. They said with RNA, you can remember apple under the tree for adenine and uracil, and car in the garage for cytosine and guanine. I thought that was really cool. So apple under the tree, A and U, and car in the garage, C and G. So that was a really nice share. I appreciate that. Thank you. Um, Let's see what else. Yeah, someone made the comment that it's a lot for the brain to retain all this information. It's good to go over all this. Yes, it is a lot for our brains to retain for sure. All right, so let's take a look at all of the possible answers here, all the correct answers. So our correct answers were A, C, and D. So let's take a look at A again. So A tells us mitosis produces 2 genetically identical cells, while meiosis produces 4 genetically unique cells. Yes, so remember that mitosis is building what I kind of collectively say our body cells, while meiosis is producing our sex cells. So we want our body cells to be identical, but we don't want our sex cells to be identical because then we would be identical to our parents and our brothers and our sisters. So in that case, we have to have genetically unique cells. And that's where processes like crossing over and independent assortment make sure that we have that unique genetic code. All right, answer C, meiosis involves 2 rounds of cell division while mitosis involves only 1. That is correct, okay? Remember that meiosis has 2 rounds because we have to get to the right chromosome number so that when sperm and egg unite, they end up with 46 chromosomes. And so we've got to go through a second division in order to get to that half number of genetic of chromosomes. All right, D was another correct one. The daughter cells in mitosis have the same number of chromosomes as the parent cell, while meiosis halves the chromosome number. Yes. So remember that mitosis' goal is to just produce an identical cell. So if we're starting with 46 chromosomes, we want to end with 46 chromosomes. That's important to mitosis because the goal of mitosis is growth and development and replacement of old cells. So mitosis, like I've often told my students when I taught, mitosis is how we get from like an almond-sized heart as a kid to a fist-sized heart as an adult. It goes through that process of having identical cells duplicated. Where meiosis, the whole goal of that is to make sperm and egg. And we've got to have the right chromosome number so that when sperm meets egg, we end up with 46 in that zygote. If we don't get to 23 in the sperm and 23 in the egg and they unite, we've got way too many chromosomes going on there. All right, let's take a look at why answer B is incorrect. So answer B says mitosis occurs in germ cells while meiosis occurs in somatic cells. So this is understanding your vocabulary. Germ cells in this context does not mean, ooh, don't touch it, it's going to get germs on you. Germ cells in this context is the broad category for sex cells. Meiosis occurs in somatic cells. Somatic cells are our body cells, everything except for sex cells. So they just have this one reversed, okay? Mitosis occurs in somatic cells. Meiosis occurs in germ cells, okay? So that was why B was incorrect. Now let's take a look at E. Both mitosis and meiosis produce gametes for sexual reproduction. No. Mitosis, body cells, meiosis, sex cells. Okay. All right, moving right along here. Now we are on to genetics, our last section. All right, which of the following can cause genetic mutations? Select all that apply. A, errors during DNA replication. B, exposure to environmental factors such as radiation or chemicals. C, proper function of DNA repair mechanisms. D, the correct pairing of nucleotides during replication. Or E, viruses that insert the genetic material into host DNA. Get those answers in there. Okay. And this one, there was actually something I didn't cover yesterday. That is a correct answer. We'll talk about it, but some of you are getting it anyways, so well done. So let's take a look here. So our correct answers are A, B, and E. So let's take a look. A, errors during DNA replication. Yes. So during DNA replication, every once in a while, something can happen. We might lose a chunk of DNA. We might get a chunk of DNA inserted. A G might end up being a T. I mean, it's just crazy, but those things happen, and that is one of the ways that a mutation can occur. B, exposure to environmental factors such as radiation or chemicals. And we hear about that often, that UV light, X-rays, things like that can cause an increased risk for those mistakes that I talked about that would occur. They're still probably going to occur during replication, but they're caused then by those environmental factors. Now, the other one that I didn't talk about yesterday that I actually will put in future ones that isn't always in TEAS, but it's really interesting to know about, is that viruses that insert their genetic material into a host DNA. So that is one of the ways that we can get a mutation. And again, it reminds me, as I was saying earlier, how creepy viruses really are, because there are viruses, retroviruses, that alter our DNA. How creepy is that, right? And I find this fascinating too. And science says the current estimate right now is that 8% of the human genome, the human DNA recipe, is viral DNA. Remnants of viruses that injected into the human DNA and then were passed on to offspring. And they came from past viruses like the Black Plague, things like that. And I find that really incredibly interesting and creepy all in one. All right, let's take a look at why C and D were incorrect. So C, proper function of DNA repair mechanisms. So when you hear that word mutation, think mistake. Okay, mutation is a genetic mistake. And so if this is talking about proper functioning, no. If proper functioning of DNA repair mechanisms are working, then we won't have a mutation. Generally, in that case, those DNA repair mechanisms actually either prevent mutations or they can kind of get rid of damaged DNA, they can fix errors. So we want that to be working properly. For D, the correct pairing of nucleotides during replication. If everything's working correctly, we're not going to have a mistake. We're not going to have a mutation. Okay. All right, moving on. Which statement best describes Mendel's law of dominance? A, each parent contributes 2 alleles for each trait and they blend together in the offspring. B, when 2 different alleles are inherited, the recessive allele is always expressed. C, a dominant allele will be expressed in an individual if at least one copy is present, while a recessive allele is only expressed if both copies are recessive. Or D, genes for different traits are inherited independently of each other and do not influence each other. Get those answers out there. All right, I think we got quite a few correct answers out there. So our correct answer is C. Mendel's law of dominance states that if an individual has one dominant allele, it's going to be expressed, while the recessive alleles are only going to be expressed if there's no dominant allele present. So remember, that's that heterozygous form. Even if one shows up, it dominates, it overpowers, and it's what we see. Okay. As far as answer A, this one's talking about them blending together. If you remember from yesterday, there are types of inheritance where there's a blend, but it wasn't Mendel's laws. Those are actually considered non-Mendelian inheritance. So that's why they don't blend. In Mendel's experience, what he worked with, one was always dominant over the other. As far as B, when 2 different alleles are inherited, the recessive allele is always expressed? No, no. So think of dominant as powerful. The dominant allele is what's going to be expressed, where the recessive is less powerful. It's not going to be expressed if there's a dominant one there. Think of personality traits, how the dominant person has a tendency to run the conversation. The dominant trait is going to be the one that's showing up. The only way that we would see the recessive is if both recessive alleles showed up and there was not a dominant trait there. Okay. As far as answer D, that's describing a different Mendel's law. That's why D was incorrect. All right, moving on. What does Mendel's law of segregation state about alleles? A, each organism has two alleles for a trait, but they separate during gamete formation so that each gamete carries only one allele. B, both alleles for a trait are passed from a parent to offspring together. C, a dominant allele will always be passed to offspring, while a recessive allele is only inherited under special conditions. Or D, the inheritance of one trait influences the inheritance of another. Get those answers out there. All right, before we expose this answer, I did have another question. Someone asked, when is a recessive allele expressed? When both alleles are recessive. So remember from yesterday's lecture, we had like big B, big B. That's a combination of both dominant alleles. We had big B, little b. That's a heterozygous form, and we would still see the dominant trait. But if we had little b, then we would see that recessive trait. And we're going to see that coming up. We have a Punnett square that we're going to go through, so that might help solidify that as well. All right, so this law of segregation, the correct answer was A. So Mendel's law of segregation says that each parent passes only one of the two alleles to their offspring because alleles separate when in meiosis. Remember that we were talking earlier about how we have to get to that 23 chromosome number. And so that's why they only get one allele, because they have half of the DNA coming from the sperm and the other half coming from the egg, so that when they unite, we have an offspring with all of the genetic information that it needs, half from mom, half from dad. That's what makes B incorrect. So B says both alleles for a trait are passed from an parent to offspring. No, then we'd end up with too much genetic information. So alleles are going to separate. C, a dominant allele will always be passed to offspring. No, while a recessive allele only under special circumstances. No, it's luck of the draw. It is literally like cards. It's just random what sperm meets with what egg, and whether it's carrying the dominant or the positive, not positive, dominant or recessive allele. And then a D, the inheritance of a trait influences the inheritance of another. No, they have no bearing. Again, it's like rolling dice. If you roll a 6 the first time on your die, there's no, that has no influence on what you're going to roll your next time. And it's kind of that same idea. Okay. All right, so which of the following best explains Mendel's law of independent assortment? Traits are inherited separately, meaning the inheritance of one trait does not affect the inheritance of another trait. B, some traits such as hair color and eye color must be inherited together. C, alleles for a single trait separate during gamete formation, ensuring each gamete gets only one allele. Or D, a recessive allele will always be masked by a dominant allele when both are present. Get those answers out there. So our correct answer here is A. Mendel's law of independent assortment states that genes for different traits are inherited independently. And what that means is, just like I talked about a rolling of a die earlier, Just because you get a 6 on the first roll doesn't mean that it influences what you get on your second roll or your third roll or your fourth roll. And that's what they're saying here too. They're saying one trait does not affect the inheritance of another. Now, one thing that Mendel didn't know about that they add kind of the caveat later is unless the genes are linked, okay? And that's something that Mendel didn't know about. And there are some traits that have a tendency to inherit together because they're really close together on the chromosome. And so depending on crossing over and independent assortment, they might end up constantly being inherited together because they're so close together on the chromosome. So that takes us back to some traits such as hair color and eye color must be inherited together. No traits must be inherited together. Even ones that are close together might have a greater probability of being inherited together, but it doesn't mean they must be. Okay. Answer C, alleles for a single trait separate during gamete formation, ensuring each gamete only gets one allele. That's true, except that's not what Mendel's law states. I mean, that's not what the question asks. Okay, that was Mendel's law of segregation. So it was a true statement, and I think sometimes that's really hard when you're taking a test to have a true statement and go, but this is right. It is right, but it isn't answering the question. And I think that's tricky. And then D, a recessive allele will always be masked by a dominant allele when both are present. Again, that is a true statement in Mendel's study of genetics. However, it doesn't answer the question. That's describing Mendel's law of dominance. Okay, and I think that can be tricky, right? So it's important that we really kind of pay very close attention to, even if it's correct, is it answering the question? All right, so now we are going to get into Punnett squares, and I will go over them, but let's see if you can do one on your own first. In a monohybrid cross between two heterozygous individuals, big F, little f, crossed with a big F, little f, what is the expected phenotypic ratio of the offspring? Look closely at that question. A, is it 1 dominant to 1 recessive? B, 3 dominant to 1 recessive? C, 2 dominant to 2 recessive? Or 1 dominant, 2 heterozygous, 1 recessive? Get those answers in. All right, you got your answers? I'm going to leave you hanging for a minute. Let's go through a cross and see what we're talking about before we go through them, okay? See if you did it the same way. So there are possible alleles. We could say that was the possible sperm, possible sperm. Oops, sorry, my bad. Sometimes it can be tricky when I have it auto-populate like this. So then over here, this could be possible egg, possible egg. We do our cross. We're bringing in our capital F's. Bringing over our capital F, lowercase f. Capital F, lowercase f. 2 lowercases, right? So now we got genotype and phenotype. So if you chose answer D, you gave us genotype. It was asking for phenotype. That's why I said read that question very closely. So what was our correct answer? It was 3 to 1. Okay. And that's what it's telling us here. Our correct answer was B. It was that 3 to 1. So you got to be really attentive to, are they asking for phenotype or genotype? Okay. 1-to-1 ratio is not going to occur in a heterozygous versus heterozygous cross. Okay. It'll occur in a heterozygous and a homozygous cross. A 2-to-2 ratio does not occur in a monohybrid cross. That would suggest different inheritance patterns. Like some of the people are mentioning on social media right now that some of these relate to other types of inheritance, and you're right. But we're talking about Mendel's form of inheritance at the moment. And then D, as I said, if you chose that, it's because you mistakenly gave us the genetic ratio, not, or I'm sorry, the genotypic ratio, not the phenotypic ratio. All right, now for those of you talking about non-Mendelian inheritance, here's one of your questions. Which of the following correctly differentiates incomplete dominance from codominance? In incomplete dominance, both alleles are fully expressed, while in codominance, the traits blend together. B, in incomplete dominance, one allele completely masks the other, while in codominance, only the dominant allele is expressed. C, in incomplete dominance, the heterozygous phenotype is a blend of the two alleles, while in codominance, both alleles are expressed simultaneously. Or D, in incomplete dominance, the recessive allele is never expressed, while in codominance, the recessive allele is only expressed if two copies are present. Get those answers out there. So our correct answer is C, okay? In incomplete dominance, the heterozygous phenotype is a blend of the alleles, while in codominance, both alleles are expressed simultaneously. That's what we saw here last night. I brought those back for us to kind of get a visual. So incomplete is that blend. Think white plus red is a mix of paint, and you end up with pink. It's a blend. Whereas codominance, like coworkers, they both show up. Okay. All right. So I had a question about the Punnett square here. How do we get to the ratios? So 3 to 1, you just drop that 4. So basically, it's just when you have fractions and both of the denominators are the same, you can drop it and you're just sticking with the numerators. 3 to 1. That's how we got to that ratio there. And it was 3 showing the dominant phenotype, 1 showing the recessive phenotype. Okay. All right. And I'm getting some great tips out on social media, and I haven't shared them because they were shared last night. So I just want to let you know I see your wonderful tips, but for time efficiency and because we had them last night, I haven't followed up with sharing them. But thank you so much. I appreciate the help. All right. So we have one now about X-linked. Why do males have a higher probability of inheriting X-linked genetic conditions compared to females? A, males have two X chromosomes, increasing their chance of inheriting the condition. B, males inherit only one X chromosome, so they do not have a second X to mask a recessive allele. Excuse me. C, females inherit only one X chromosome while males inherit two. Or D, X-linked conditions are only passed from father to son, increasing male inheritance rates. Get those answers out there. Okay, for anyone on Zoom, I put out in the chat list just a little reminder for incomplete and codominance to help you. That was a question I was getting several on, and I thought I would, just for time efficiency, put it there. All right, so this idea of males having a higher probability of X-linked genetic conditions. Our correct Answer is B. Males only have one X chromosome because remember, that's what determines whether you're male or female. If you get two X's, you're female. If you get an X and a Y, that makes you male. Okay. So if that trait sticks on the X chromosome, they only get one shot at getting that condition. Okay. And we're going to take a look at that. So we're going to kind of save this rationale till we go through the Punnett square, and then we'll come back to this rationale. So let's do a practice. If we have an X-linked cross and we have a female and she's heterozygous for the trait. So notice we've got our X chromosomes, but we're using a superscript to show that it's linked. Okay. So that means she has one dominant component or one allele for the trait, and she has one recessive for the trait, crossed with a male that is dominant for the trait, and it doesn't sit on the Y chromosome, it sits on the X chromosome. So let's take a look at what this looks like in a Punnett square. There's our female. There's our male. There's our first cross. See how we're bringing them over? There's our second cross. Notice that these are females. Now there's our first male. That first male doesn't have the condition if it's sitting on the recessive trait, which is typically the case. This male has. So what is that really telling us? Well, let's look at our genotype and our phenotype. First of all, our genotype. We have 1 out of 4 females that are unaffected. That's this person right here. Or the probability of it. Make sure you understand these are just probabilities. We have 1 female, what they refer to in a situation like this as a carrier. Yes, she's heterozygous, but she's carrying the allele for this trait right there, just like mom was a carrier. Then we have 1 male unaffected right there. 1 male affected. So what does that really mean phenotypically? Well, it means that 100% in this cross with this male and this female, their female children will not express the condition, because even if she's a carrier, she's got a dominant trait to mask it. She's got a dominant allele to cover it up. But look at here, 50% of the males, we got a 50-50 shot with the males. He's either not going to have it or he's going to have it. All right. Okay, so looking at our rationale here, that's what made B correct. Males inherit only 1 X chromosome, so they do not have that second X to mask that recessive allele. Okay, so let's take a look at A. Why was A wrong? Males have 2 X chromosomes. That makes it wrong right there. You could stop reading, because you know to be male, you have to have an X and a Y. C, females inherit only 1 X chromosome. You could stop right there, not even keep reading, because we know that to be female, you have to have 2 X's. D, X-linked conditions are only passed from father to son, increasing male inheritance rates. No, actually, if we look closely, in order to be male, where did that Y come from? That Y came from dad. In order to be male, where did that Y come from? It came from dad. The X comes from mom. Okay? All right, let's see where we're at here. We have wrapped it up. So at this point, I always like to bring back to our screen here our QR codes for the free live webinars, to rewatch webinars, our question bank, our on-demand videos, tutoring, our packages like I talked about at the beginning. We also can be found on all the socials. You can get tutoring if you'd like. And of course, I would love some feedback. Tell me how I did today. Tell me what you think of our new format. Lecture one day, questions the next. Okay? Great spending the afternoon with you. Give me some feedback. Hope you have a lovely day, and I will look forward to seeing you. I think my next one coming up is chemistry next week. Okay? Take care.